This video article describes a procedure for obtaining glycosylation profiles of 20 different glycoproteins in a hepatocellular carcinoma patient serum sample using a chemically blocked antibody microarray. First, glycoprotein-specific antibodies are printed directly onto microarray slides, and antibody glycans are blocked to prevent lectin binding. When patient serum samples are applied, glycoproteins from the sample are captured by the antibodies. Biotinylated glycan-binding proteins are then added to detect the glycans, and di-conjugated neutravidin is added to label the biotinylated lectins. The microarray slide is then scanned to detect fluorescence. Analysis of the resulting data reveals the glycosylation profiles of the sample proteins.
This technique has several advantages over existing methods like HPLC. This method allows for detection of individual glycoproteins in their native status. It is faster and easier to screening glycosylation alteration for multiple proteins simultaneously.
Right. We found that biomarkers of disease, particularly of cancer and liver cancer, which we've looked at most carefully, are often glycosylated. And the ability to detect specific glycoforms we found greatly enhances biomarkers' performance in its sensitivity and in its specificity.
So this method can provide insights into glycosylation alterations on multiple serum proteins in HCC. It can also be applied to the pathology study.